This majestic landscape is the boreal forest of the southwestern Yukon near Kluane Lake. This northern region is predicted to experience dramatic climatic shifts over the next 50 years, with temperatures warming and precipitation patterns likely to change drastically. As the boreal forest warms, all organisms in the ecosystem will most likely be affected in some manner. These effects have begun to be understood and documented for some species, but many questions still remain regarding how the boreal forest will respond to climate change, as is the case for so many ecosystems on Earth. An extremely important but often overlooked component of this forest ecosystem is the microbiome of the soil. The function of this microbial ecosystem is essential to the overall health and productivity of the boreal forest. Bacteria play major roles in nutrient cycling here, while fungi are the primary decomposers in this cold, nutrient-poor soil landscape. In addition, fungi form important symbiotic relationships with plant roots, which aid in nutrient acquisition of these plants and promote above-ground productivity. Soil microbes play a major role in carbon storage in these soils, and their response to climate change may ultimately determine whether the boreal forest remains an important global carbon sink or becomes a source of carbon emissions contributing to warming on our planet. My name is Francis Quinby, and I'm a PhD student at Trent University. I'm researching the effects of climate change on the soil microbial ecosystems of Canada's western boreal forest, and conducting my fieldwork in the Kluane Valley of the southwestern Yukon. For my research, I've designed and built a field experiment near Kluane Lake, simulating warming and variable precipitation patterns for soil plots within white spruce forest stands. I built my experiment in forest stands on site at the Kluane Lake Research Station, which is on the southern shore of Kluane Lake. The experiment is designed as a 2x3 factorial study with 4 replicates. The temperature factor has 2 treatment levels, consisting of a control and a warming treatment, for which I've used open-top chambers, which act as a sort of greenhouse without a top. Precipitation was a 3-level treatment in which a third of the plots received an increased volume of rainfall, a third receive a decrease, and the remainder receive a control. To ensure that precipitation was received in a consistent manner, all plots were covered with clear fiberglass material, and I applied the appropriate amount of water to plots using a commercial sprayer following each rainfall event. I collected soil samples this past summer at the beginning of the experiment, and I'll collect soil samples again at the end of the study. By then comparing soil properties in the bacterial and fungal communities present within these soils, I'll determine how the climatic factors manipulated in my experiment affected the soil biotic and abiotic characteristics. Soils are extremely biodiverse, and hundreds to thousands of species of bacteria and fungi can be present in a single sample. By extracting the microbial DNA from soils, amplifying specific genomic regions using PCR, and then subjecting this product to next-generation metabarcoding sequencing techniques, I'll be able to determine the composition of the bacterial and fungal communities. By performing this methodology on soils collected from the beginning and at the end of my experiment, and comparing the results, I'll be able to assess how these communities shift as a result of the simulated climatic changes. I'm very fortunate to have the opportunity to conduct my research in such a beautiful and pristine northern landscape. But there are definitely a number of logistical challenges when building a climate change experiment in such a remote area. For example, I had to carry all my materials to field sites by hand, as there were no roads to directly access my plots. I actually ended up using some standing dead trees, which I felled and cut to length, to construct the base of my experimental precipitation exclusion covers in order to minimize the costs associated with lumber and materials. Luckily, the management at the Kluane Lake Research Station were extremely helpful in providing me with the use of tools and repurposed lumber, and for this I'm especially grateful. I'm immensely grateful to the Weston Family Foundation for supporting my research through this fellowship, as well as for supporting numerous other important conservation research projects. Without this support, many aspects of these research efforts would not be possible.